If you happen to stand in an IVF lab early in the morning, right before the chaos starts, there is this very distinct hum from the incubators.
Oh, absolutely. It's a very specific frequency. You never really forget it.
Right. And when you are just standing there, poring over the patient files, prepping for the day, you really feel the immense weight of what is about to happen. Because, in most fields of medicine, you expect this degree of mechanical certainty.
Sure, like if a bone breaks, you take an x-ray, you see the jagged fracture, and the orthopedic surgeon knows exactly what physical intervention to do.
Exactly. But you, the clinical embryologist, you operate in a totally different universe of decision making. You sit down at that microscope and you are asked to make judgments that will quite literally alter the generational trajectory of an entire family.
It is a staggering level of responsibility, you
Hmm?
know?
It really is. Every major call you make in that lab, which oocyte is competent enough to inject, which zygote actually merits extended culture, or which blastocyst ultimately goes into the transfer catheter, it fundamentally relies on your assessment of a single microscopic organelle. You are basically banking everything on the
You
nucleus.
really are. And we often fall into this trap of viewing the nucleus as just a simple passive storage locker for DNA, like a biological hard drive or something.
Right. Just a bag of
Yeah,
genes.
exactly.
Yeah.
But it is so much more dynamic than that. I mean, it is the command center, it's the architectural scaffold, and it is the definitive developmental compass for every single embryo you evaluate. Without a highly coordinated, flawlessly functional nucleus, the massive epigenetic reprogramming required for early development just simply cannot occur.
To tip it and see basically becomes impossible.
Exactly. And ultimately, there is no life.
So for those of you who are gearing up for the ESHRE certification exam, the stakes are essentially twofold. You carry the clinical weight in the lab every day, but you also face this examination board that absolutely expects you to possess a masterful mechanistic understanding of reproductive cell biology.
Yeah, you cannot just memorize what a good embryo looks like. They want you to understand the molecular why behind the morphological criteria you were using at the bench.
And that is our mission for this deep dive today. We are taking a really comprehensive journey into the nucleus. We are going to start at the absolute outer perimeter and work our way down into the deepest, most hidden architecture of the genome.
Because we have to connect the invisible biophysics of the cell to the macroscopic clinical realities you deal with, every single day.
Right. And we need to approach this with the exact rigor those ESHRE standards demand. We're going to dissect the biochemistry, the physical forces, the molecular machinery governing everything from oocyte maturation to day one pronuclear scoring.
Because the exam doesn't just test if you can spot an abnormality. It tests your ability to explain the underlying cellular failure that actually caused it.
That sets our trajectory perfectly, I think. So
Oh,
before
yeah.
we even consider the DNA, we have to look at the vault protecting it, the nuclear envelope.
Oh, the envelope.
Yeah, for a long time, my own mental model of the envelope was basically just a simple single-ply plastic bag holding a soup of genetic material.
Which is how it's taught in basic bio.
Right. But structurally, it is a highly specialized double phospholipid bilayer.
The architecture is entirely deliberate. The cell constructed a double-walled fortress. And between those two walls lies a fluid-filled gap we call the perinuclear space.
Which is not just some empty buffer zone, right?
No, not at all. It is a critical functional compartment.
Let's break down that outer wall first.
Yeah.
The outer nuclear membrane, or the ONM. From a purely spatial perspective, this outer membrane is actually continuous with the rough endoplasmic reticulum.
Yeah, that always blows my mind a little bit.
Same. If you follow the lipid bilayer, you just seamlessly transition from the nuclear envelope straight into the ER network. And it is studded with ribosomes on its cytosolic surface, which effectively integrates the nucleus into the cell's broader protein manufacturing system.
Right, and that continuity with the ER is essential for synthesizing the proteins that will eventually live in the nuclear envelope itself. But beyond just protein synthesis, the outer nuclear membrane serves this profound mechanical function.
It's physically bolted down, isn't it?
Exactly. It is bolted to the cell's cytoskeleton through a network of specialized transmembrane proteins called LINC complexes.
LINC standing for linker of nucleoskeleton and cytoskeleton. So if I'm picturing this mechanically, the outer membrane has these protein anchors that reach out into the cytoplasm and grab onto the actin filaments and microtubules.
They do. And the biophysics of that connection are just fascinating. I mean, the LNC complex is essentially a protein bridge. You have these cache domain proteins spanning the outer membrane, binding to the cytoskeleton outside. And then they reach completely across the paranuclear space and bind to SUN domain proteins on the inner membrane, which in turn bind to the nuclear lamina on the inside.
Wait, so it creates an unbroken mechanical cable running from the cytoplasm straight through the double membrane directly into the genome.
That is exactly what it does.
That implies something incredible about the physical environment of the IVF lab. Because of this continuous cable, the outer nuclear membrane has to be acutely sensitive to the physical environment.
Oh, highly sensitive.
Right, so when an embryologist is handling cells, say, the sheer stress of pipetting an oocyte for denudation, or the physical pressure of the ICSI needle during injection, those mechanical forces are actually being transmitted.
Yeah, they pull on the cytoskeleton, which tugs on the LINC complexes, which literally stretches the nucleus and the DNA inside.
Mechanical force is a biological signal.
It absolutely is. Mechanotransduction is a massive field of study right now. And it shows that physical tension on the nucleus can physically alter chromatin organization and change gene expression. We really have to remember that our physical manipulations in the lab are not happening in a vacuum. They register as tension along those LANC complexes.
That is a very humbling thought. You are quite literally pulling on the DNA with your pipette.
You are.
Now, what about the inner wall of this fortress, the inner nuclear membrane?
So the inner nuclear membrane faces the nucleoplasm, and its protein composition is completely distinct from the outer membrane. It is packed with integral proteins whose primary job is to anchor the interior architecture.
So they are binding to the nuclear lamina to hold the shape of the nucleus.
Yes, and they also provide tethering points for the chromatin itself, which, as we'll get into, plays a massive role in regulating which genes are active and which are silenced at the periphery.
And trapped between these two very distinct walls is that paranuclear space. Now, since the outer membrane is continuous with the ER, I assume this space between the membranes is continuous with the lumen of the ER as well.
Yes, the paranuclear space and the ER lumen form a single continuous fluid compartment. This is what allows lipid molecules and certain newly synthesized membrane proteins to flow freely between the ER and the nuclear envelope.
Which guarantees the nucleus always has the materials it needs to repair its boundaries and grow.
Exactly, which is especially critical during those rapid cell cycles of early cleavage stage embryos.
But a double-walled fortress with a fluid moat is entirely useless if nothing can get in or out.
Right, you just starve the cell.
Exactly. The cell needs to import fuel and machinery, and it needs to export the products of transcription. Which brings us to the gatekeepers, the nuclear pore complexes, or NPCs.
Calling them gatekeepers almost understates their scale. The nuclear pore complex is an absolute behemoth of a structure. We are looking at a protein assembly weighing roughly 120 megadaltons. It's one of the largest, most elaborate molecular machines in the entire eukaryotic cell.
120 megadaltons is really difficult to conceptualize. So
Yeah.
for comparison, a standard ribosome is around, what, 3 to 4 megadaltons?
Roughly, yeah.
So a single nuclear pore is essentially the mass of 30 or 40 ribosomes fused together.
It is colossal. And these complexes are inserted exactly at the sites where the inner and outer nuclear membranes curve inward and fuse with one another. Each individual pore is constructed from about 30 different core proteins, which we call nucleoporins or NUPs.
And they assemble in this stunning 8-fold rotational symmetry, right? It looks like a massive 8-spoked molecular wheel embedded in the membrane.
It really does. But the logistics of the traffic flowing through that wheel are what's staggering. Picture the nucleus of an actively dividing blastomere. You have entire armies of histones flooding in from the cytoplasm to package the newly replicated DNA.
Right, and transcription factors,
Yep.
massive DNA polymerases, ribosomal proteins, all fighting their way inside.
Exactly. And simultaneously, you have messenger RNA, transfer RNA, and newly assembled ribosomal subunits trying to flood out into the cytoplasm.
How does a single pore handle a busy two-way biochemical highway like that without everything just jamming together?
Well, it handles it by strictly separating the traffic based on physical size and biochemical signaling. So for very small molecules ions, simple sugars, small metabolites, basically anything under a molecular weight of roughly 40 kilodaltons, the pore is essentially just a passive sieve.
So those tiny molecules just diffuse down their concentration gradients right through the aqueous channel in the center of the pore. No energy required.
Just simple fluid dynamics. But as you know, an mRNA transcript bound to all its export proteins is vastly larger than 40 kilodaltons. A DNA polymerase is huge.
Passive diffusion would take millions of years for macromolecules that size to cross the membrane.
Exactly. And that is the mechanical problem the cell solves with active signal-mediated transport. The center of the nuclear pore isn't just an empty pipe. It is filled with these specialized nucleoporins that contain extensive repeats of the amino acids phenylalanine and glycine.
The FG repeats.
Right, the FG repeats. And they form this dense, hydrophobic gel, almost like molecular kelp, just blocking large molecules from passing through.
So how on earth does a massive protein melt through a hydrophobic kelp forest?
It requires a molecular escort. If a large protein needs to enter the nucleus, it must possess a specific amino acid sequence built right into its structure, called the nuclear localization signal, or NLS.
It's quite literally a zip code.
Exactly, it's a zip code. So specialized carrier proteins out of the cytoplasm called importance recognize that NLS zip code. They bind to the cargo protein and they interact chemically with that FG gel. The important momentarily melts the hydrophobic mesh, pulling the cargo right through the pore.
And I assume the reverse process works for getting things out.
Yeah, export relies on a very similar logic. The cargo has to carry a nuclear export signal, an NES. And this is recognized by a different class of carriers called exportins, which ferry the cargo from the nucleoplasm out to the cytoplasm.
But this kind of directed against the gradient transport, that violates the laws of thermodynamics unless you pay for it. Active transport requires a massive energy currency.
Oh, it's very expensive. The currency and the directional compass for all of this traffic is managed by a small enzyme called RAN-GTPase. RAN exists in two different states depending on what it is bound to, RAN-GTP or RAN-GDP.
Okay.
And the cell invests heavy amounts of energy to maintain a very steep gradient. There is a high concentration of RAN-GTP strictly inside the nucleus and a high concentration of RAN-GDP strictly out in the cytoplasm.
Let me make sure I have the mechanics of this gradient right. This gradient is what tells the importans and exportins which way they're supposed to go, right?
Right.
So when an important brings a cargo into the nucleus, it encounters that high level of RAN-GTP. The RAN-GTP binds to the important, and that forces it to drop its cargo inside.
You have the biophysics exactly right. The binding of RAN-GTP acts as the physical release mechanism for imported cargo. Conversely, for export, an exportin can only bind its cargo inside the nucleus when RAN-GTP is also present to lock the complex together.
Oh, I see.
Yeah, and then once that whole complex moves out to the cytoplasm, the GTP is hydrolyzed to GDP, the complex falls apart, and the cargo is finally released into the cell body.
Now, why does a clinical embryologist studying for the ESHRE exam need to memorize the biochemistry of RAN-GTPase and exportins? Where does this actually hit the clinical bench?
It hits the bench at the very beginning of the timeline, specifically with oocyte competence. Think about the growth phase of the germinal vesicle oocyte inside the follicle. The oocyte is actively transcribing a staggering volume of maternal messenger RNA.
Right, because the early embryo does not transcribe its own genome for the first few days. It runs entirely on the maternal mRNA stockpiled by the oocyte.
Precisely. So all of that maternal mRNA has to get out of the GE nucleus and into the cytoplasm to be stored. If the nuclear pore complexes fail to efficiently export that mRNA using these NES and exportin pathways, or if the RAN gradient is somehow compromised due to metabolic stress in the oocyte, that maternal stockpile is never built.
And we see this clinically, don't we?
We do. We see it as an oocyte that may look morphologically completely normal. It might even mature and fertilize, but then it abruptly arrests at the cleavage stage because it simply ran out of fuel before embryonic genome activation could even occur. The pore's efficiency directly dictates the developmental competence.
That is fascinating. Okay, so moving inward from the pores, lining the entire inner face of the inner nuclear membrane, we find a dense, highly organized protein meshwork. This is the nuclear lamina. It feels like the rebar of the nucleus.
Rebar is an excellent analogy. The lamina is the structural scaffold, and its components are members of the type V intermediate filament protein family, which we collectively call lamins. And we categorize them primarily into two groups based on their physical properties: the A-type lamins and the B-type lamins.
Right. The A-type lamins, specifically lamins A and C, which are actually alternative splice products of the exact same gene, they provide the mechanical stiffness. They give the nucleus its rigidity and its ability to withstand those compressive forces from the cytoplasm we discussed earlier.
Yeah, and complementing that stiffness, you have the B-type lamins, lamins B1 and B2. These are anchored more permanently to the inner membrane, and they provide essential elasticity and overall structural integrity. But beyond just holding the balloon open, so to speak, lamin B proteins also play a surprising extended role during cell division by actually helping to orient the mitotic spindle.
But the lamina is not just inert structural scaffolding. If I remember correctly, it actively participates in organizing the genome. It literally grabs sections of the DNA.
It does. The lamina serves as a massive tethering surface for the chromatin. Specific, really massive stretches of the genome are physically anchored to the lamina. We call these regions lamina-associated domains, or LADs.
LADs, okay.
And the biochemical rule of thumb here is critical. When a segment of DNA is plastered against the nuclear lamina in a LAD, it is almost always heterochromatic. It is tightly packed, it's gene poor, and it is transcriptionally silenced.
It is essentially sent to the periphery to be ignored. By anchoring it to the wall, the cell keeps those genes away from the active transcription factories in the center of the nucleus.
The spatial organization dictates the function. When a stem cell begins to differentiate and needs to activate a new set of genes, those specific genes physically detach from the lamina. They migrate deeper into the nuclear interior, they unfold, and they become transcriptionally active. The lamina is a very dynamic manager of epigenetic geography.
That brings up a fascinating point regarding embryology. The gametes are highly specialized, terminally differentiated cells. But after fertilization, they have to become totipotent again. The embryo has to be able to become anything. So do the lamins stay the same during that transition?
They absolutely do not, and this is a heavily tested concept on the exam. Gametes have specialized lamina architectures. The mammalian oocyte, for instance, expresses a unique oocyte-specific lamin called lamin B3 within the germinal vesicle.
Oh, I see.
But immediately following fertilization, as the zygote begins its journey toward the blastocyst stage, the composition of the nuclear lamina completely remodels. The embryo literally tears down the gamete-specific scaffolding and constructs a somatic-type lamina.
The physical architecture of the nucleus has to be completely rebuilt to support the new totipotent identity. That is the physical reality of epigenetic reprogramming.
Exactly.
Since the lamina is busy silencing massive sections of the genome, let's look closer at the DNA itself. Let's explore chromatin states, because this answers one of the most fundamental questions in biology. If every cell in the body has the exact same DNA sequence, how does a granulosa cell know to act like a granulosa cell, and a sperm cell know to act like a sperm cell?
The secret lies entirely in accessibility. Chromatin, which is the functional complex of DNA wound around its histone proteins, exists in different states of compaction. The genome is effectively partitioned into two broad categories based on just how tightly the DNA is folded.
The open accessible state is euchromatin.
Right, euchromatin is physically loose. Because it is unspooled, the DNA sequence is physically exposed to the nucleoplasm. Transcription factors, RNA, polymerases, all the regulatory machinery can easily bind to the promoters. Euchromatin regions are typically gene-dense, highly transcriptionally active, and usually positioned toward the interior of the nucleus.
And from a biochemical standpoint, how does the cell mark a region to stay open like that?
The cell attaches specific chemical tags to the tails of the histone proteins. The hallmark epigenetic signatures for active euchromatin include the trimethylation of histone 3 at lysine 4, often abbreviated as H3K4Me3, and the acetylation of histone 3 at lysine 27, or H3K27AIC.
We have to unpack the biophysics of that acetylation because it is just so elegant. Lysine is a positively charged amino acid, and DNA has a negatively charged phosphate backbone, so naturally they stick together tightly like magnets.
Exactly.
But when the cell adds an acetyl group to that lysine, it neutralizes the positive charge. The magnetic grip fails, and the DNA naturally loosens and pushes away. That is the actual mechanical why behind euchromatin.
That thermodynamic shift is the engine of gene expression. But on the opposing side, we have heterochromatin. This is chromatin that is deeply compacted, heavily condensed, and practically invisible to the transcription machinery. It is the silenced genome, often shoved out to the periphery at those LADs we just discussed.
Heterochromatin isn't just one monolithic thing, though. We divide it into constitutive and facultative heterochromatin.
Right. Constitutive heterochromatin represents the regions of the genome that are permanently silenced in almost every single cell type. We're talking about the structural regions, centromeres, telomeres, and vast tracts of highly repetitive non-coding DNA. The cell marks these permanently locked regions with a different tag, histone 3 lysine 9 trimethylation, or H3K9E3.
And that methylation acts as a docking pad, right?
Yes, it acts as a docking pad for specialized proteins that condense the DNA into this impenetrable block.
And facultative heterochromatin.
Facultative heterochromatin is much more flexible. These are genes that are turned off right now in this specific cell, but might need to be turned on later, or they're active in a totally different tissue type. The classic textbook example of facultative heterochromatin is the inactive X chromosome in female mammalian cells.
The bar body.
Exactly. One entire X chromosome is crumpled down into a silenced bar body, which is heavily harmed by histone 3 lysine 27 trimethylation H3K27A3.
This molecular deep dive is fascinating, but how does an embryologist sitting in a dark lab staring down the oculars of a light microscope actually use this information? Can you literally see euchromatin and heterochromatin at the bench?
You can, and interpreting it is a core competency for the ESHRE exam. When you examine an immature germinal vesicle, or GV, oocyte under high magnification, the spatial arrangement of the chromatin inside that massive nucleus is a direct visual readout of its epigenetic state. We categorize GVs into two primary chromatin configurations.
The surrounded nucleolus configuration and the non-surrounded nucleolus configuration.
Right. Let's look at the surrounded nucleolus, or SN, configuration first. Under the microscope, you see the chromatin highly condensed into a tight, distinct ring that completely encases the nucleolus. From a molecular perspective, this immense block of heterochromatin indicates that the oocyte has largely finished its massive transcriptional phase.
It has stockpiled all the maternal mRNA it needs.
Exactly. And it's just shutting down transcription to prepare for the violent physical movements of meiosis. Clinically, SN oocytes demonstrate high meiotic competence and superior developmental potential.
That makes perfect sense. The silencing is a sign of readiness. What about the other configuration?
The non-surrounded nucleolus, or NSN configuration. Here, the chromatin does not form a dense ring. Instead, it is dispersed loosely and diffusely throughout the nucleoplasm. This implies the DNA is still largely euchromatic and actively transcribing.
So the oocyte is still desperately trying to build its maternal reserve.
Right, it is not ready. If you force an NSN oocyte to undergo in vitro maturation, it generally shows significantly lower fertilization rates and a much higher propensity for early embryonic arrest.
You are quite literally grading the epigenetic maturity of the oocyte through a microscope lens. It's a beautiful intersection of molecular biology and clinical observation. But all of that chromatin, whether it's SN or NSN, eventually has to pack down incredibly tightly to survive the mechanics of cell division. It has to transform into the discrete rod-like structures we recognize on a karyotype, chromosomes.
Yes, the heavy lifting of division.
Let's review chromosome anatomy and classification.
Well, for the vast majority of the cell cycle, during interphase, chromosomes don't look like discrete objects at all. They exist as that complex, tangled territory of chromatin we just described. They only hypercondense into discrete, transportable structures as the cell actually enters division. At this stage, each chromosome consists of two identical sister chromatids, replicated during S phase, and bound tightly together at a primary constriction point known as the centromere.
And the centromere divides the chromosome into two distinct arms. The shorter arm is always designated the P arm, which stems from the French word petite. By convention, when we draw or map a chromosome, the P arm goes on top. The longer arm is designated the Q arm, simply because Q is the next letter in the alphabet after key, and it sits on the bottom.
Right. And the fundamental classification system for human chromosomes relies entirely on the physical location of that centromere.
Let's walk through the shapes. If the centromere sits dead in the middle, creating a P arm and a Q arm of roughly equal length, we call that a metacentric chromosome. If the centromere is shifted slightly off-center, making the P arm visibly shorter than the Q arm, that is submetacentric.
Correct.
And if the centromere is pushed drastically toward one end, leaving a tiny, almost vestigial P arm consisting mostly of a small stalk and a satellite, that is an acrocentric chromosome.
And theoretically, if the centromere is located at the absolute terminal tip of the chromosome with no P arm at all, it is telocentric. However, it is a crucial piece of trivia for genetic pathology that normal human karyotypes do not contain telocentric chromosomes. Mice have them, but humans do not. We operate with 46 chromosomes in our diploid somatic cells, 22 pairs of autosomes, and 1 pair of sex chromosomes. Our gametes are haploid, carrying 23, and the elegant fusion of syngamy restores the diploid state.
I want to pull focus back to the acrocentric chromosomes for a moment. In the human genome, the acrocentrics are chromosomes 13, 14, 15, 21, and 22. These 5 specific chromosomes share a unique and vital structural feature on their tiny p arms. They house the nucleolar organizer regions, or NORs.
The NORs are massive tandem repeats of the genes that code for ribosomal RNA. Because the p-arms of these 5 acrocentric chromosomes are packed with these highly homologous repetitive NOR sequences, they are notoriously prone to structural errors during meiosis.
And this is the biomechanical root of Robertsonian translocations. Because the tiny p arms of chromosomes 13, 14, 15, 21, and 22 are so genetically similar with all that rDNA, they frequently undergo non-homologous recombination.
They get confused.
Right. 2 acrocentric chromosomes will accidentally fuse at the centromere, losing their short arms entirely and creating one massive combined chromosome.
And that structural anomaly is one of the primary reasons patients present to an IVF clinic requiring pre-implantation genetic testing for structural rearrangements, or PGTSR. A patient carrying a balanced Robertsonian translocation may be perfectly healthy themselves, but their gametes face a catastrophic risk of severe aneuploidy.
This really highlights how dependent we are on the mechanical perfection of segregation. To pull those chromosomes apart accurately during division, the cell utilizes the ultimate molecular towing engine, which is built right onto that centromere. Let's delve into the centromere and the kinetochore.
This is where the biophysics of division become incredibly complex. We intuitively assume that the centromere, this crucial primary constriction point, must be defined by a very specific hard-coded sequence of DNA, but it isn't. The centromere is defined epigenetically.
That is always a shock to learn. You could take the exact same DNA sequence and move it elsewhere, and it wouldn't act like a centromere.
The location of the centromere is dictated by the presence of a specialized histone variant called CENPA. In the nucleosomes that make up the centromere chromatin, standard histone H3 is entirely replaced by CENPA. This CENPA protein acts as a permanent, self-propagating epigenetic marker. When the cell divides, it actively replenishes CENPA at that specific locus, maintaining the centromere's identity entirely independent of the underlying DNA sequence.
So CENPA is the foundational landing pad. And once the cell commits to division, an enormous multi-layered protein machine called the kinetochore is constructed directly on top of that CENPA pad.
Right, the kinetochore is the true interface between the chromosome and the mitotic or meiotic spindle. It is composed of well over 100 different proteins, organized into 2 distinct physical domains. The inner kinetochore binds fiercely to the CENPA chromatin, creating a structural anchor, and the outer kinetochore extends outward and dynamically interacts with the plus ends of the spindle microtubules.
The outer kinetochore is where the physical force is generated. It houses motor proteins, including cytoplasmic dynin and various kinesins, which literally walk along the microtubules, generating the tremendous pulling force required to rip the sister chromatids apart during anaphase.
But generating force is only half the battle. If the kinetochore just blindly pulled, you would have catastrophic missegregation. The outer kinetochore also houses what is arguably the most critical surveillance system in the cell cycle, the spindle assembly checkpoint, or SAC.
The SAC operates precisely like the safety interlock systems on an amusement park ride. Imagine a roller coaster. The ride operator cannot pull the lever to start the ride until every single passenger has pulled down their safety lap bar until it clicks. If even one lap bar is loose, the central computer registers a fault, and the ride is mechanically locked in the station.
That is the exact logic of the SAC. The checkpoint machinery, driven by proteins like MAD2, constantly monitors every single kinetochore. It is checking 2 parameters. First, is a microtubule physically attached? And second, is there bi-orientated tension, meaning, are the microtubules pulling from opposite poles with equal force?
And if they aren't?
If a kinetochore is unattached or lacks tension, the SAC complex actively inhibits a protein called CDC20, which in turn halts the anaphase promoting complex. The cell is chemically locked in metaphase. The ride cannot start.
The cell will patiently wait until that last stray chromosome is captured by the spindle. But this safety mechanism isn't flawless, and its failure is the defining pathology of reproductive aging. Let's discuss the maternal age effect.
This is a cornerstone concept for the ESHRE curriculum. We know that as a woman ages, the aneuploidy rate in her oocytes rises exponentially. The primary mechanistic reason for this is the degradation of the spindle assembly checkpoint. As oocytes sit arrested in the ovaries for decades, The abundance of key SEC proteins slowly depletes.
The lap bars on the roller coaster get faulty.
Exactly. An older oocyte might have a chromosome that is only attached to one side of the spindle or not attached at all. But because the SAC proteins are depleted, the checkpoint fails to detect the error. It gives the green light, anaphase proceeds, and both sister chromatids are pulled into the same cell, resulting in an aneuploid gamete. The mechanical failure of the SAC is the driving force behind the age-related decline in oocyte competence.
So we have explored the mechanical center of the chromosome. Now let's travel to the extreme periphery, to the very ends of the DNA strands, where a completely different kind of biological clock is ticking. Telomeres and telomerase.
The linear nature of eukaryotic chromosomes creates a severe structural vulnerability. A naked, exposed end of double-stranded DNA looks identical to a catastrophic DNA double-strand break. If the cell's damage repair machinery detected a naked chromosome end, it would immediately attempt to fuse it to another chromosome, causing massive translocations, or it would trigger apoptosis to kill the cell.
To mask those ends and prevent the cell from self-destructing, we cap the chromosomes with telomeres. In humans, these are long, non-coding, highly repetitive sequences consisting of the bases TTG repeated over and over for thousands of base pairs.
But the sequence alone isn't enough. It requires a specialized physical architecture to hide the end. A group of 6 specialized proteins, collectively known as the sheltering complex, binds to these TTT repeats. The sheltering complex actually grabs the overhanging single strand of DNA at the very tip and physically folds it backward, burying it into the double-stranded DNA further up the telomere. This forms a structure called a T-loop.
It's like taking the frayed end of a rope and weaving it back into the main braided cord so the end is completely hidden. The damage sensors scan the chromosome, see the smooth T-loop, and move on.
It is a brilliant architectural solution, but it introduces a new, insurmountable problem for normal somatic cells, the end replication problem. Conventional DNA polymerases can only add nucleotides to a pre-existing strand. They require an RNA primer to begin. When the replication fork reaches the absolute end of the lagging strand, there's no physical space left to place a final primer. Consequently, the polymerase cannot copy the very last segment of the DNA.
So with every single cell division, the daughter cell inherits a slightly shorter chromosome. You lose roughly 50 to 200 base pairs of telomeric sequence every time the cell divides.
Over a lifetime, those TKG repeats are whittled down. Eventually, the telomere becomes critically short. There is no longer enough sequence to form the protected T-loop. The sheltering complex falls off, the naked DNA is exposed, and the cell recognizes a critical damage signal, forcing it into replicative senescence or programmed cell death. The telomere is the cell's absolute biological clock.
Unless the cell possesses the enzyme that can turn back the clock telomerase.
Telomerase is a fascinating molecular machine. It is a ribonucleoprotein, meaning it is an enzyme that carries its own internal piece of RNA, known as TERK. This RNA acts as a template. Telomerase functions as a specialized reverse transcriptase, binding to the shortened chromosome end and using its own RNA template to actively synthesize and add fresh TTD repeats back onto the telomere.
Now, most of the specialized somatic cells in an adult human turn off the telomerase gene. They accept their finite lifespan. But stem cells, and specifically germ cells, maintain highly active telomerase to preserve their replicative potential across generations.
That preservation is why telomeres are heavily researched in the context of reproductive medicine. Studies consistently show that shortened telomere length in oocytes and surrounding granulosa cells correlates strongly with diminished ovarian reserve, lower fertilization rates, and impaired embryo development. The oocyte's telomere length serves as a quantifiable biomarker of its molecular age, which frequently diverges from the patient's chronological age.
We have spent a lot of time discussing these massive macrostructures of the chromosome, but we need to zoom all the way down to the atomic level to understand how you actually construct a chromosome in the first place. The packaging math is almost unbelievable. A single human diploid cell contains over 2 meters of linear DNA, and the nucleus is roughly 6 micrometers in diameter.
The spatial compression required is staggering. You have to pack a thread of DNA that is hundreds of thousands of times longer than the container it sits in, without allowing it to form a catastrophic, unbreakable knot.
And the fundamental unit that makes this packaging possible, the very first spool the DNA winds around, is the nucleosome.
The nucleosome relies on a core complex of proteins called histones, specifically the histone octamer. The cell takes 2 copies each of 4 distinct core histones, H2A, H2B, H3, and H4, and assembles them into an 8-part protein spool.
The biophysics of how the DNA binds to the spool is beautifully simple. Those core histones are exceptionally rich in two specific amino acids, lysine and arginine. At physiological pH, lysine and arginine carry a strong positive charge. DNA, due to the phosphate groups running along its entire backbone, carries a heavy negative charge. It is basic electrostatic attraction. The negative DNA is magnetically drawn to wrap tightly around the positive histone spool.
It wraps precisely 146 base pairs of DNA around the octamer in approximately 1.7 left-handed superhelical turns. This specific geometry forms the nucleosome core particle, but a spool of thread will just unravel if you don't secure the end. To lock the DNA in place, a fifth type of histone, the linker histone H1, binds to the exterior of the nucleosome at the exact points where the DNA strand enters and exits the spool. Histone H1 acts like a molecular clamp, securing the coil and pulling adjacent nucleosomes closer together to facilitate higher-order stacking.
This elegant packaging system applies to almost every cell in the body. But embryologists need to be acutely aware of one massive, dramatic exception, and it happens during spermatogenesis.
The mammalian sperm cell is an extreme example of biological minimalism. To create a highly hydrodynamic, motile cell capable of delivering a payload to the oocyte, the sperm nucleus must achieve a level of compaction far beyond what standard histones can provide. During the late stages of spermiogenesis, the developing sperm cell aggressively strips out nearly 85 to 90% of its core histones.
It tears down the nucleosomes. What does it replace them with?
It replaces them with protamines. Protamines are small, incredibly basic proteins that are overwhelmingly composed of arginine, giving them a ferocious positive charge. Even more importantly, protamines are rich in cysteine residues. Once the protamines bind the DNA, these cysteines form extensive disulfide cross-links with neighboring protamines, essentially locking the entire paternal genome into an incredibly dense, almost crystalline state.
The DNA is practically armor-plated. But this creates a massive biological problem at the moment of fertilization. The early embryo's transcription machinery cannot read DNA that is locked in a protamine crystal.
That is the precarious challenge of fertilization. When the sperm head enters the ooplasm, the oocyte must execute a flawless rapid rescue mission. The oocyte utilizes its stored pools of glutathione to chemically reduce and break those rigid disulfide bonds in the protamines. The zygote must then rapidly evict the protamines from the paternal DNA and replace them with maternal histones stored in the cytoplasm.
It's an entire genomic remodel happening in a matter of hours. If the oocyte is oxidatively stressed and lacks glutathione, or if the machinery to swap the protamines fails, the paternal genome remains locked in its crystalline state. The pronucleus will fail to form properly, and the zygote will inevitably arrest. This paternal chromatin reprogramming is a highly tested vulnerability.
Let's assume the histones are correctly placed. The DNA wrapped around the nucleosome is only the very first level of compaction. We need to walk through the hierarchical packing that scales up to form the full nucleus.
If we start with the naked DNA double helix, that fiber is about 2 nanometers wide. Wrap it around the histone octamers, and you get the classic beads on a string formation, which is about 10 nanometers wide. Add the linker histone H1 to clamp it down, and the nucleosomes coil upon themselves to form a dense 30-nanometer chromatin fiber. I visualize this like a coiled retro telephone cord, but a telephone cord is still a linear string. How do we organize it spatially?
The 30-nanometer fiber is next gathered into massive architectural structures known as chromatin loops. The cell manages these loops using specialized motor proteins belonging to the SMC family, specifically the cohesin and condensin complexes. Cohesin is essentially a ring-shaped protein that binds to the DNA. It acts like a molecular winch.
A winch, okay.
Through an energy-driven process called loop extrusion, the cohesin ring reels the DNA fiber through its center, creating an ever-expanding loop until it hits specific stop sign proteins, typically CTCF, which anchor the base of the loop.
These loops aren't just random piles of DNA. They are highly organized regulatory neighborhoods called topologically associating domains, or TADs.
TADs are the functional units of the genome's 3D architecture. A single TAD essentially fences off a local neighborhood of DNA. Within that specific loop, enhancer sequences can freely bump into and activate the promoter regions of their target genes. But crucially, the boundaries of the TAD prevent those enhancers from accidentally activating genes in the neighboring loop. It isolates gene regulation into discrete physical zones.
And when you zoom out even further, looking at the entire nucleus, these TADs and loops gather together to form chromosome territories.
Exactly. We used to picture the interphase nucleus like a bowl of spaghetti with chromosomes randomly tangled together. We now know that is completely false. Every single chromosome occupies a distinct, non-overlapping, strictly defined volume within the nucleus, a chromosome territory. They have specific zip codes.
But that beautiful, meticulously organized 3D architecture is incredibly fragile. It does not survive cell division.
It cannot survive division. As the cell prepares to enter mitosis, the interphase architecture is systematically dismantled. The cohesin rings are removed, the TAD boundaries dissolve, and a different SMC complex condensin takes over to aggressively pack the chromatin into the dense 700 nanometer thick mitotic chromatids that can withstand the physical violence of the spindle pulling them apart. Once cytokinesis is complete, the new daughter cells face the monumental task of rebuilding their entire 3D chromosome territories and TAD structures completely from scratch.
To understand how a newly formed cell rebuilds that architecture, we have to look at how the real estate within those chromosome territories is zoned. And the genome organizes itself into two very broad functional spatial zones, the A compartment and the B compartment.
This is a macro-level view of transcriptional geography. The DNA physically segregates itself based on its activity level.
Let's look at the A compartment first. I think of this as the cell's main reading room.
The A compartment is euchromatin dense. It contains the highly active, gene-rich regions of the genome. Structurally, the regions of DNA assigned to the A compartment tend to physically cluster together, usually migrating toward the interior volume of the nucleus, where the concentration of RNA polymerases and transcription factors is the highest.
Conversely, the B compartment is the archive stacks.
The B compartment is dominated by heterochromatin. It is gene-poor, tightly compacted, and generally silent. These B compartment regions naturally avoid the A compartment regions. They tend to physically migrate toward the periphery of the nucleus, associating heavily with the nuclear lamina to form those lamina-associated domains, the LADs, that we discussed earlier.
That spatial segregation implies a rigid destiny. If a gene is assigned to the B compartment archive, is it locked there permanently?
No, and that fluidity is the cornerstone of cellular development. The compartments are not permanent jails. Cells utilize a process called compartment switching. When an embryonic stem cell begins differentiating into, say, a neural progenitor, it must drastically alter its gene expression. To accomplish this, massive stretches of its DNA literally unspool from the B compartment at the nuclear periphery, migrate into the central A compartment, and become active. Simultaneously, genes that are no longer needed migrate out to the periphery to be silenced in the B compartment.
Does this compartment switching happen during fertilization?
It happens in the most extreme, dramatic fashion possible during fertilization. Remember, the mature oocyte and the mature sperm possess highly specialized, terminally differentiated chromatin architectures. When they fuse to form the zygote, the embryo must achieve absolute tutapancy. To do this, both the maternal and paternal genomes must globally erase their previous spatial organizations. The zygote undergoes a massive reorganization of its A and B compartments, physically resetting the architecture to a tutipotent state. If this spatial remodeling is incomplete or aberrantly regulated, the embryo will fail to activate its genome correctly and will arrest early in cleavage.
The physical location of a gene dictates its fate. Now, so far, we've focused almost entirely on the DNA and the membranes enclosing it. But the nucleus is an incredibly busy factory, and it needs subcompartments to manage the massive workload of transcribing RNA, assembling ribosomes, and splicing mRNA. But here's the architectural paradox. The nucleus has no internal lipid membranes to create separate rooms. How does it organize this work?
It organizes the work by constructing nuclear bodies. These are distinct, highly concentrated functional organelles floating within the nucleoplasm, but as you noted, they lack any lipid membrane boundary whatsoever.
If they don't have a membrane, how do they keep their components from just diffusing away into the nucleoplasmic soup?
They're held together by a biophysical phenomenon known as liquid phase separation.
It's the classic salad dressing analogy, oil and water spontaneously separating into distinct droplets based on their chemical properties, creating a sharp physical boundary without needing a physical wall.
That is the exact thermodynamic principle at play. Inside the nucleus, you have specific proteins, often intrinsically disordered proteins, and specific RNA molecules that have a strong chemical affinity for one another. When their local concentration hits a critical threshold, they spontaneously condense out of the nucleoplasm, forming a distinct, dense liquid droplet. This concentrates the biochemical reactants, massively increasing the efficiency and speed of their specific reactions.
The absolute heavyweight champion of these phase-separated droplets is the nucleolus.
The nucleolus is the largest, most conspicuous structure inside the interphase nucleus. And it forms precisely around those specific chromosomal regions we highlighted earlier, the nucleolar organizer regions, the ESHREs, located on the tiny p-arms of the acrocentric chromosomes.
The acrocentrics converge, and the nucleolus phases into existence around them. Because the nucleolus has one crucial, life-sustaining job, it is the cell's ribosome factory. It aggressively transcribes ribosomal RNA from those NORs, processes the RNA, and assembles it with ribosomal proteins to forge the pre-ribosomal subunits that are eventually exported through the nuclear pores.
And we cannot overstate how clinically paramount the nucleolus is for the early embryo. When the oocyte matures, it shuts down transcription. But once fertilization occurs and the embryonic genome activates a few days later, the embryo is going to need to aggressively synthesize its own proteins. To do that, it desperately needs an army of new ribosomes. The health, size, and functional assembly of the nucleolus in the early cleavage stages are quite literally matters of life and death for the embryo.
Beyond the nucleolus, there are several other critical membraneless bodies. We have Cajal bodies, which serve as recycling and assembly centers for the spliceosome machinery, the machines that edit messenger RNA. There are PML bodies, which act as signaling hubs during DNA damage responses. And nuclear speckles, which are essentially storage droplets of splicing factors conveniently located right next to highly active euchromatin domains.
The absence of internal membranes allows the nucleus to be incredibly dynamic. These nuclear bodies can assemble in seconds when needed, scale up their size based on the transcriptional demand, and dissolve instantly when the cell enters mitosis.
This entire deep dive has been operating on a fundamental, unstated assumption that every single cell contains exactly one nucleus. But biology is never that rigid. We need to explore nuclear variants. What happens when a cell has no nucleus at all, or when it has far too many?
Variation in nuclear number is usually a deliberate, highly evolved adaptation to solve a specific physiological problem. Take the enucleate cell, a cell entirely devoid of a nucleus. The quintessential example in mammalian biology is the mature erythrocyte, the red blood cell.
As the red blood cell develops in the bone marrow, it reaches a point where it literally ejects its own nucleus, permanently discarding its genome.
It sacrifices its genetic command center for strict mechanical utility. By ejecting the nucleus, the erythrocyte maximizes its internal volume, allowing it to pack in significantly more hemoglobin. More importantly, without that large, rigid organelle, the cell becomes incredibly flexible, capable of physically folding itself in half to squeeze through the microscopic lumens of peripheral capillaries. Platelets operate on a similar principle. They are merely enucleate fragments sheared off from massive megakaryocytes.
The ultimate trade-off, of course, is that an enucleate cell has absolutely no capacity for transcription. It cannot repair itself, and it cannot divide. Its lifespan is strictly finite, about 120 days for a red blood cell, and then it must be destroyed and replaced.
On the opposite end of the spectrum, we have intentional multinucleation. Skeletal muscle fibers, for instance, are massive, syncytious, single, continuous cellular tubes formed by the fusion of hundreds of individual precursor cells, containing hundreds of distinct nuclei sharing a single cytoplasm.
A single nucleus simply could not generate enough mRNA to service a cell of that physical length. And in reproductive biology, we see an even more extreme example in the placenta, the syncytia trophoblast.
The syncytia trophoblast is arguably the most critical tissue in pregnancy. It forms the outermost epithelial layer of the placental villi, directly interfacing with maternal blood. It is a massive, continuous, multinucleated sheet with no internal cellular boundaries. This unique architecture prevents maternal immune cells from simply squeezing between cell junctions to attack the fetus. The vast number of shared nuclei are required to drive the astronomical transcription rates needed to mediate all nutrient exchange and to synthesize massive quantities of critical hormones like HCG and progesterone. In that context, multinucleation is functionally beautiful.
But, context is everything. What about when we encounter multinucleation isolated in the IVF lab? You are sitting at the scope doing a day 2 or day 3 cleavage stage check. You focus through a 4-cell embryo, and inside one of those individual blastomeres, you clearly see 2 distinct nuclei.
That is an entirely different biological reality. In an early cleavage stage embryo, multinucleated blastomeres are a sign of severe pathological cellular failure.
Mechanistically, how does a blastomere end up with 2 nuclei?
It typically stems from a failure of the cytoskeletal mechanics. The cell successfully duplicates its DNA and completes nuclear division karyokinesis. But the actin ring fails to contract, meaning the cell never undergoes physical cytoplasmic division or cytokinesis. You are left with one massive cell containing 2 complete diploid nuclei. Alternatively, The spindle assembly checkpoint may have failed, leading to lagging chromosomes that get packaged into their own separate, smaller micronuclei.
It is a massive red flag for the embryologist.
A profound red flag. The joint ESHRE and Alpha-Kinskay consensus guidelines mandate that embryos exhibiting multinucleated blastomeres must be heavily penalized in grading. Why? Because extensive clinical data demonstrates that a blastomere displaying multinucleation has a drastically higher incidence of complex aneuploidy. It indicates a fundamental catastrophic breakdown in cell cycle regulation. While some embryos can occasionally self-correct or sequester a single multinucleated cell, generally speaking, an embryo with multinucleation is deprioritized for transfer because its implantation potential is severely compromised.
This is the perfect pivot. We are going to tie all of this dense structural molecular biology directly into the strict chronological timeline of the IVF lab. We need to walk through the perilous nuclear journey of oocyte maturation.
The sequence of events is the most highly scrutinized timeline you will monitor. You are observing an oocyte that has been arrested in prophase of meiosis since fetal development, potentially arrested for 30 or 40 years, finally receiving the signal to resume division and become a mature, fertilizable egg.
It begins at the germinal vesicle stage, the GV. Under the microscope, the GV is an unmistakable massive nucleus, roughly 25 micrometers in diameter. It has a prominent nucleolus and, as we detailed earlier, a fully intact double membrane envelope. And if you use specialized optics, you might even discern whether the chromatin is in that competent SN configuration or the less competent NSN configuration.
The first critical milestone in maturation is germinal vesicle breakdown, or GVBD. In vivo, this dramatic structural collapse is triggered by the mid-cycle LH surge. In our in vitro maturation protocols, simply isolating the oocyte from the inhibitory environment of the surrounding follicle cells can trigger the cascade.
What is the biochemical trigger that actually shatters the envelope?
The master regulator is a kinase complex known as MPF, or maturation promoting factor, which consists of CDK1 bound to cyclin B. When the inhibitory signals are removed, MPF activity surges. This kinase aggressively phosphorylates the nuclear pore complexes, causing them to rapidly disassemble. Crucially, MPF also phosphorylates the nuclear lamina, causing those lamin filaments to instantly depolymerize. Without its scaffold, the double lipid envelope shatters into tiny vesicles, and the highly condensed chromatin is dumped directly into the cytoplasm as the cell officially resumes meiosis.
The envelope is completely gone, and the oocyte organizes those chromosomes on the first meiotic spindle. It has reached the M1 stage. Now, this is a dangerous pitfall for junior embryologists. An oocyte at the M1 stage is not a mature egg.
Absolutely not. At the M1 stage, the homologous chromosomes are aligned on the metaphase plate, but they have not yet segregated. The cell is halfway through the division. You cannot, under any circumstances, perform ICSI on an M1 oocyte. Injecting a sperm now would result in catastrophic chaotic ploidy.
You must wait until the division finishes and the oocyte reaches M2.
Maturation is only achieved when the oocyte reaches the M2 stage. At this point, the homologous chromosomes have separated, and the oocyte has ejected exactly half of its genetic material into a small, tightly packaged waste disposal unit, the first polar body, which sits in the paravitelline space. The oocyte then halts again, arresting precisely on the second meiotic spindle, waiting for the trigger of fertilization to complete the final division.
So, practically, when an embryologist is stripping the cumulus cells and grading an oocyte for ICSI, they're visually hunting for the presence of that first polar body and assessing the clarity of the cytoplasm to confirm the M2 stage. But we must remember the invisible vulnerability we've been discussing. At M2, there is no nuclear envelope protecting the genome.
That is the ultimate mechanical vulnerability in the IVF lab. At the M2 stage, the chromosomes are entirely reliant on the fragile web of spindle microtubules holding them perfectly aligned just beneath the plasma membrane. The nuclear envelope is gone. Spindle integrity is the only thing preventing aneuploidy.
And those microtubules are notoriously unstable.
Highly unstable. They are exquisitely sensitive to temperature fluctuations. If you leave an M2 oocyte out of the incubator on an unheated stage, and its temperature drops even a few degrees towards room temperature, those spindle microtubules will begin to depolymerize in a matter of minutes. The spindle literally dissolves. If you then inject that oocyte, or even if it recovers and rebuilds the spindle later, the chromosome segregation during that final division will be highly prone to errors. Temperature control during M2 handling is the most rigid rule in embryology for this exact molecular reason.
It's a high-wire act with no safety net. Okay, let's assume we maintain perfect conditions. We have a beautiful M2 oocyte, we successfully perform ICSI, and we place it safely back into the incubator. We wait. Fast forward 16 to 18 hours. The next morning, we return to the microscope to check the zygote. This is pronuclear assessment, and it relies entirely on everything we've discussed today.
Early pronuclear assessment is arguably the most information-rich, non-invasive evaluation an embryologist will perform. We're looking directly at the morphological reconstruction of the two genomes.
To set the scene, the sperm has entered the euplasm. The oocyte was triggered to finish meiosis, extruding the second polar body. The sperm chromatin has been stripped of its progamines and repacked with histones. Now, the maternal and paternal genomes do not just instantly crash together, do they?
No, they maintain their separate architectural boundaries for many hours. Each genome constructs its own complete nuclear envelope, forming two distinct pronuclei, or PN, within the shared cytoplasm. They will coexist independently, each undergoing massive DNA replication and that critical epigenetic compartment switching we discussed earlier before their envelopes finally dissolve and the chromosomes intermingle at syngamy to form the first mitotic spindle.
So we look down the microscope at 17 hours post-insemination. What are the hallmarks of a normal, highly competent zygote?
A normal fertilization event yields a 2PN zygote. The first criteria are geographic. You want to see exactly two pronuclei that have migrated to a central position within the cytoplasm, and they should be of approximately equal size. But you cannot stop at the outer boundary. You must focus through the pronuclei to evaluate their internal structures.
We are specifically looking for the nucleolar precursor bodies, the NPBs. These appear as dense, highly refractive spheres floating inside the pronuclei. And based on our earlier discussion about phase separation and NORs, these are literally the precursor droplets that will eventually fuse to form the embryo's critical nucleolus factory.
Exactly. The spatial distribution and the number of these MPBs are highly predictive of the embryo's developmental trajectory. The ideal, most morphologically favorable pattern, often categorized as a top-tier Z-score, is when both pronuclei contain roughly equal numbers of MPBs, and critically, those MPBs are polarized.
Polarized meaning they have all migrated and lined up right at the internal junction where the two pronuclear envelopes are touching each other. Why is that specific alignment so crucial?
Polarization indicates that the internal cytoskeletal mechanics, the microtubulasters, and the internal chromatin organizations of both pronuclei are perfectly synchronized. They are mechanically aligning their genomes in preparation for a seamless syngamy. When you see unequal numbers of MPBs or NPBs scattered randomly throughout the nucleoplasm like buckshot, it strongly correlates with asynchronous development and has significantly lower potential to reach the blastocyst stage.
The geometry tells the story, but an embryologist also has to instantly recognize the abnormal patterns.
Yes, the pathological states.
Yeah.
If you look in and see a 1PN zygote, you face a dilemma. It could represent parthenogenetic activation, where the oocyte mechanically activated in response to the ICSI needle, but the sperm failed to decondense. Or it could mean both genomes decondensed but prematurely fused into a single massive nucleus. In either scenario, the ploidy is completely unverified, and a 1PN is generally considered unsuitable for transfer.
And if you see a 3PN?
A 3PN zygote indicates the presence of three distinct pronuclei. In standard IVF, this most commonly results from catastrophic polyspermy 2 sperm penetrating the zona pellucida simultaneously. In ICSI, since we control sperm entry, a 3PN almost always indicates a maternal meiotic failure. The oocyte failed to extrude its second polar body, leaving the entire maternal diploid genome trapped inside, which then forms two maternal pronuclei alongside the single paternal pronucleus. Again, this is a triploid embryo and cannot be transferred.
And a 0PN simply means no pronuclei are visible. This could mean complete fertilization failure, but there is a timing catch, isn't there?
Yes, the timing window is critical. A 0PN at 17 hours might mean failure, or it could mean you are looking at a hyperfast developing embryo where the pronuclear envelopes have already broken down and sygamy has already occurred. You must leave it in culture and observe whether it cleaves normally over the next 24 hours.
This precise evaluation is why formal scoring methodologies, like the Scott system or the Z-score, are so heavily emphasized in the ESHRE curriculum. They remove the subjectivity. They rely on the absolute physical geometries of the nucleus size, equality, centrality, and NPB polarization. Early pronuclear assessment gives us a window into the molecular soul of the embryo long before it ever begins to cleave.
It is incredibly satisfying to realize that the most complex, abstract concepts of molecular biology, phase separation, histone modifications, microtubule dynamics, perfectly align with the strict, practical clinical guidelines you utilize at the bench.
Let's pull all of this together and summarize the incredible anatomical journey we've mapped out today.
We began at the outer perimeter, exploring the double-walled nuclear envelope. We saw how the outer membrane mechanically links to the cytoskeleton via LINC complexes, while the inner membrane anchors the silent genome. We navigated the sheer scale of the nuclear pore complexes, detailing the energy-driven RAN-GTPase-dependent import and export cycles that are vital for preserving the oocyte's maternal mRNA stockpile.
We looked at the nuclear lamina, not merely as a structural tentpole, but as an active repressor of lamina-associated domains, noting how it radically remodels itself immediately after fertilization to support epigenetic reprogramming. We broke down chromatin into the accessible acetylated euchromatin and the silenced methylated heterochromatin and connected that directly to the SN versus NSN grading in germinal vesicle oocytes.
We examined the extreme mechanical engineering of the chromosome. We tracked the epigenetically defined CENPA centromere and the massive kinetochore that houses the crucial spindle assembly checkpoint. Understanding how the age-related degradation of that checkpoint is the primary driver of maternal aneuploidy. We looked at the T-loop architecture of telomeres and the preserving power of telomerase.
We scaled the mathematical miracle of DNA packaging, from the charge base wrapping around the histone octamer and the perilous protamine swap required by the sperm, all the way up through cohesin-driven loop extrusion, TADS, and into the massive spatial territories of the active A compartment and the silenced B compartment.
We explored how phase separation creates membraneless functional zones, like the crucial ribosome-producing nucleolus. We addressed the pathology of multinucleated blastomeres, and finally, we applied every bit of the structural biology to the strict timeline of the IVF lab, from the MPF-driven breakdown of the GV, the temperature-sensitive vulnerability of the M2 spindle, right through to the predictive NPB geometries of the 2PN zygote.
It is a massive volume of structural and molecular biology, but as we've seen, every single nanometer of this architecture dictates a direct clinical outcome. Which brings us to our final thought. We have spent an hour discussing how the zygote miraculously tears down the specialized chromatin structures of the gametes and rebuilds them to achieve totipotence. But look toward the horizon of reproductive medicine, in vitro gamegenesis, or IVG. The idea of taking a patient's simple skin fibroblast, reverting it to a stem cell, and chemically coaxing it in a petri dish to become a fully mature, competent oocyte.
It is the ultimate frontier. We can already force a skin cell to express the right genes. But the true barrier to IVG isn't just turning genes on or off. The barrier is architectural. To create an artificial gamete that actually works, we don't just have to copy the sequence, we have to synthetically engineer the correct spatial 3D folding. We have to artificially construct the exact lamina-associated domains, perfectly segregate the A and B compartments, and build a flawless M2 spindle all outside the body.
Will we ever be able to artificially synthesize the epigenetic architecture of life from scratch? That is the question the next generation of embryologists will have to answer. The next time you sit down at your rig and you look down that microscope at a oocyte or a zygote, I want you to remember what you are actually seeing. You're not just looking at a circle on a screen or assigning a morphological grade. You are witnessing the physical manifestation of billions of microscopic structural decisions. You are watching the architecture of life rearranging itself in real time.
Understanding the depths of nuclear structure is not just a hurdle you must clear to pass the ESHRE exam. It is the very language of clinical embryology. When you truly understand the nucleus, you can finally understand the embryo's intent.
Thank you for joining us on this deep dive. Carry this mechanistic foundation back into the lab with you tomorrow and let it change the way you evaluate every single cell. Until next time.
Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.